All right, so we're going to move into chapter 17 today. And so chapter 17 is primarily about two things. Nucleic acids, which is kind of the fundamentals of DNA, and protein synthesis, which is basically what your DNA is meant to do. And so we'll focus on primarily the first kind of bits today, looking at what nucleic acids are, how DNA is put together, and then we'll also get a little bit into replication, like how your body recreates your genetic code, which is happening in millions of your cells right now. And that's where we'll call it for today. And then on next Tuesday, we'll get into DNA transcription, which is how we take that genetic material and actually use it to create proteins. And those proteins are what are used by your body to do most of your biological functions. So when we start with this first part in chapter 17, we have to understand what makes up a nucleic acid. So nucleic acids represent kind of the last of those biomolecules that are in your body. So we started with carbohydrates, we talked about lipids, we talked about proteins, now we're talking about nucleic acids. And all nucleic acids have three components to them. There is the base. This is the part of the nucleic acid that is kind of unique and different. So there are 4 bases for DNA. There are 4 bases for RNA as well. 3 of those bases are held in common between the 2. One of them is different. There is a sugar molecule. The sugar molecule is unique. DNA has a different sugar compared to RNA. And then there is a phosphate group. And this phosphate group is pretty much the same all of this. But if I were to look at DNA, what I would see is that every single DNA would have these 2 components on it. They'd have the phosphate group, it would have deoxyribose as the sugar. For RNA, we would have the phosphate group, we would have ribose as the sugar. The only thing that would be different amongst DNA or amongst RNA acids would be the identity of that base. Now, the bases come in 4 different forms. Again, there are 4 bases for DNA: guanine, cytosine, adenine, and thymine. There are 4 bases for RNA, and they are pretty much the same, except thymine doesn't exist in RNA. It's replaced by a different base called uracil. The nucleic acids are known primarily by their 1-letter abbreviations. A for adenine, G for guanine, C for cytosine, and T for thymine. Uracil, which again is only in RNA and replaces the thymine in the code, is given by the letter U. Now, these bases fall into 2 different kind of forms. There are what are called the pyrimidine bases. The pyrimidine bases have this nitrogen ring. So kind of imagine a benzene ring with a couple of nitrogens on opposite sides. And that would be pyrimidine. And then with purine, you've got that same pyrimidine base, but we now have a second ring attached to it. So the ones that are purine are your adenine and your guanine. So your A and your G. Your pyrimidine would be the cytosine and your thymine. So your C and your T. And since uracil replaces thymine, it is going to be similar in structure to that pyrimidine. And so looking at this structurally speaking, you can see why thymine and uracil, even though they're not exactly the same thing, you can see why uracil is able to kind of insert itself in the absence of thymine, because really the only difference between these two structures is that methyl group on this position right next to the top carbonyl on the left. Whereas with adenine and thymine, again, or I'm sorry, adenine and guanosine, these are purines, and there are some relatively significant differences between. So these are the 5 bases. And again, we don't want to get confused with thinking of these as bases in the pH kind of sense. What we're talking about here is these these are bases in terms of this is the base unit for a DNA nucleic acid. So that's the first component. The second component of a nucleic acid is a pentose sugar. Pentose 5, this is a 5-carbon sugar, makes a 5-membered ring with, again, if we think about what those Hayworth structures are showing us here, we've got a 5-member ring in the ribose here. What that means is, so here's ribose. Ribose is the sugar backbone of RNA. The R in RNA stands for ribose. So this is a ribose nucleic acid, RNA. D in DNA stands for deoxyribose. So that's a deoxyribose nucleic acid. The difference in structure is on this second carbon. So again, remember how we numbered these Hayworth structures. The carbon immediately to the right of the oxygen in the ring is carbon number 1. And so we can see that deoxyribose has no oxygen on that second carbon. That is what makes DNA different from RNA, other than the thiamine uracil change out. So the reactions that take place to turn this sugar, ribose, and this base into what is called a nucleoside. We're going to be doing another reaction where we have water as a byproduct. So this is not an esterification reaction or even a peptide. This is just simply what is called a dehydration reaction, where these are the hydrogen and the hydroxyl groups here are going to come off to form the water, and the carbon off of the ring is going to bind itself to the nitrogen in this base. That's what's going to create a nucleoside. Since we're dealing with a sugar reaction, this is why it's called a glycosidic bond. Again, deriving itself from the carbohydrate base, which is similar to the kinds of linkages that we saw when we were talking about carbohydrates specifically. And this is an N-glycosidic bond because it's bonding through a nitrogen as opposed to a carbon. So here's our adenosine, the combination of the ribose with the sugar base there, or I'm sorry, not the sugar base, with the nucleic base there. So by definition, a nucleoside is this chemical combination of sugar and base. And we get this from this dehydration reaction, where again, the hydrogen off of the base and the hydroxyl group off of the either the ribose or the deoxyribose, they're gonna jump off and make water. And the leftover bond is going to form between this first carbon here and the nitrogen in the base. And so this would become, so this is adenosine. This would be the adenosine that you would find in RNA. Because again, we've got a full-on ribose here as opposed to deoxyribose. So that's a nucleoside. And we want to be real careful about our names here, because a nucleoside sounds a lot like a nucleotide, and nucleotide is basically this, and now we add the phosphate group. So a nucleotide takes that nucleoside, which again was the combination of the sugar and the base, and now we attach that phosphate group to it. And the phosphate group always attaches to this fifth carbon that's standing up on top of the ring. And so it is nucleotides that are ultimately going to be linked together to create a nucleic acid. So very similar to what we saw with proteins, where we had the amino acids that we linked together to make protein. Here, we're going to have the nucleotides that we link together to make a nucleic acid. And that nucleic acid will start as kind of a chain, and then we'll start to twist on itself and give us what we are most familiar with as, what DNA looks like. So again, bringing up our nucleotide, it has three components. It has the phosphate group, which we just added at the end. It has the sugar group, which when combined with the base, either the pyrimidine or the purine, will give us a nucleoside. The nucleoside plus the phosphate makes a nucleotide. I know I'm throwing a lot of alphabet soup at you right now. So try to bear with me. And again, if you got questions about stuff, stop me. We've got the time. So just as a comparison here, DNA versus RNA. Lots and lots of common features between them. They're all nucleic acids at their heart. When we look at the base, when we look at the bases, practically the same bases. A, C, and G are common to both. The difference between DNA and RNA is whether we use thymine or uracil. The sugar backbone is different in both because, again, DNA is the deoxyribose, whereas RNA is just the ribose. And all that is different is what that second carbon has on it. Does it have an OH group or does it have two H's on it? But the definitions of nucleoside and nucleotide are practically the same. I'm taking that chemical base, whatever it is, and I'm attaching it to the sugar. That makes a nucleoside. I take that base and that sugar, and I throw on a phosphate group, and that becomes a nucleotide. And the nucleic acid is just a polymer, a end-to-end train of one nucleotide attached to another, attached to another, attached to another. And we make up a long chain. So let's just make sure that we understand some of our terms here. Here I have an adenine and cytosine. One of these is a purine base. One of these is a pyrimidine base. Which one is which? So show me with your fingers. 1 for A, 2 for B. Which one is the purine base? Okay, so again, it's a definition thing. We have to know the difference between purines and pyrimidines. Pyrimidines are the one ring, purines are the double ring. So purines being the double ring, the adenosine is the double ring, which means that the pyrimidine is the cytosine, which is B. Now we can get into kind of looking at things from a structural standpoint. So here's where things can get a little bit more messy on our side. Because now we are going to start linking these together to create a chain. And unfortunately for us, the chain that we are creating is not as simple as just linking one of these sugars to the next, carbon to carbon to carbon to carbon. We saw that in sugars when we were making polysaccharides. It was relatively easy to kind of follow along because they always seem to go in the same place. You know, this first carbon to this fourth carbon, this first carbon to this fourth carbon, or 1 to 6, 1 to 6, 1 to 6. They always seem to work out the same way. Here, these phosphate groups don't quite behave the same way. The phosphate doesn't attach to just one of the sugars. It actually attaches to 2 sugars. Got a phosphate attachment here to this sugar here at the 5th carbon, and we've got a attachment to this sugar at the 3rd carbon. And what that creates is we've got a backbone that's going to be a lot less flexible because we've got now attached in multiple places instead of one. But it also creates something that we saw in proteins as well, the idea of terminuses. So if you remember from proteins, we had the nitrogen terminus and we had the carbon terminus. And because of the way that we always drew it, we always knew which side was which. Nitrogen was on the left, carbon was on the right. Well, here we have a 3 terminus and a 5 terminus. And why are they called 3 and 5? Because those are the carbons that are left hanging off at the ends. So in this bottom sugar, we can see that this carbon right here currently does not have a phosphate group on it. And it's the 3rd carbon in that ring. So that's the free end on the 3rd carbon. And we refer to this as the 3 terminus. Or the 3 terminal. And over here, we can see that this 5th carbon has a phosphate group on it, but it does not have another sugar connection there. This is on the 5th carbon. So this is the 5 terminus or the 5 terminal. And this can be kind of confusing because when we see that prime, that apostrophe mark, that mark has a lot of meaning depending upon different kind of ideas. For me, I always got it confused. I always wanted to think of it in feet because single apostrophe is shorthand for feet in measurement, just like double is inches. So I always go, okay, well, what's going on at the 3-foot end? What's going on at the 5-foot end? Obviously, that's not correct. DNA are not 3 feet and 5 feet long. Thank goodness for that. But at the same time, that's what we're setting up here. There's a 3 end and a 5 end. And depending upon how the DNA is drawn, it's either going to be kind of top and bottom like this one is, or it's going to be left and right. And so you just got to pay attention to the notation here, because they'll usually give you at least some kind of clue about it. The reason that's important is because these kinds of nucleic acid sequences don't exist as just simply a strand. There's a complementary string that is on the opposite side of it. And it is set up in exactly the opposite direction. So if the 3-terminal is on the left, on the one strand, it's going to be on the right on the complementary strand. It's going to appear backwards to each other. So similar to proteins, we talk about this linkage of nucleic acids one after another. We refer to that as the primary structure. Just like with amino acids, we refer to the primary structure as just one amino acid linked after another. Same thing with these nucleotides. The sequence of nucleotides from one to the other, to the next to the next. That's the primary structure of this nucleic acid. And just like we saw with proteins, there are these terminal ends. Now in proteins, it was the N-terminal and the C-terminal. Here it's the 3-terminal and the 5-terminal. Now, the links that hold each of these together are known as phosphodiester bonds. And that sounds really complicated. But if we break it down, it's actually kind of, we can figure it out. Phospho, because it is this phosphate group that is doing the connecting. And diester, because what we are seeing here is this phosphorus double bonded to oxygen is acting in the same way that a carbonyl would if it was carbon. If I put carbon in that spot instead of phosphorus, we would be talking about esters, because we'd have a carbonyl on one side and we'd have oxygens on either side next to them. Well, carbon can't make 5 bonds. Phosphorus can. And in doing so, that phosphorus is going to create 2 places where it can make these bonds. And these bonds are more similar to ester bonds than they are to ether bonds. And so we've got the phosphorus center, so that's the phospho part. And we've got these 2 ester bonds. And so rather than saying 2 ester, we're going to use that di prefix to stand in for 2. So I've got a phosphodiester bond, a phosphorus making 2 ester linkages, one to the first sugar group and a second to the next sugar group in the nucleic acid sequence. So now, why do they call it 3,5? Because that's the carbon pair that they're linking up. They're linking up the 3rd carbon from that first sugar, the one on top, with the 5 carbon on the next sugar, the one that's on the bottom. And the nice thing about this is that that's the only kind of linkage that gets made between the nucleotides and each other. That phosphodiester doesn't bounce around. It's always from the third carbon of one sugar to the fifth carbon of the next. And then the third carbon of that sugar to the fifth carbon of the next sugar. And then the third carbon of that sugar to the fifth carbon of the next one. And that's how we build this backbone of sorts. When it comes to sequencing, just like what we saw with proteins, we will also use a similar kind of notation for sequencing nucleic acids. We just list off the nucleotides in order. Unlike with proteins where we had, sometimes we could use the 3-letter abbreviations and sometimes we could use the 1-letter abbreviations. With DNA, there's only 1-letter abbreviations. A, T, G, C, or A, U, G, C. It's RNA. And we just list out the order. So our nucleic acid is traveling this way from top to bottom here. And so our first nucleotide is adenine, so that's the A. Then cytosine, that's the C. Then guanine, that's the G. And then in this case, since we are talking about RNA, we've got a uracil group here, that would be the U. So our nucleic acid is going from the 5' end to the 3' end. Okay. What is the term that belongs in the blank here? It is phosphodiester. It's our 15-point Scrabble word of the day. It is phosphodiester bonds that link together the ribose sugar groups, whether they be ribose or deoxyribose. All right, which base is at the free 5-phosphate end? Okay. If by A we mean option A, yes. So remember, we go from 5 to 3 as we go from left to right. So the free phosphate end on the 5 terminal, I almost said 5 foot there, I stopped myself, is cytosine, which is A. Yeah. Multiple choice gets a little bit funny when we get into nucleic acids because A and C are both actual nucleic acids as nucleotides. Okay, so that's the primary structure. That's how we line them up and stack them out and put them in a row. The last bit that we want to get into today is how does it actually look? Because we know from our earliest biology experiences in school, we know that DNA is not just a long strand of stuff. We know that DNA looks like this, classic double helix. But how do we get from these chains, long linear stretches, to this double helix? Well, the answer is kind of twofold here. First of all, just like with proteins, we have to consider that there are going to be interactions between the pieces of that backbone and each other. We're going to see the formation of helices, kind of like a secondary structure in a protein. The other thing that we're going to have to look into is the fact that no DNA is just a single strand. It's actually a double strand. So how do I know what that second strand looks like? Well, luckily we've got some research to help us. Going back to the 40s, the 1940s, it was discovered at the time we could actually analyze genetic material and determine its composition, its makeup. And what we found out through these analyses was that when we looked at DNA, the amount of adenine and the amount of thymine was always the same. And the amount of guanine and cytosine was always the same as well. And that seemed kind of odd, because that would mean that it isn't quite as random as maybe we were once thinking. What was even more interesting was we were able to determine from these data and from some other work, and this is eventually where the story will turn to Watson and Crick, Rosalind Franklin, and some of the other names that you're probably at least somewhat familiar with in the history and story of DNA. But what we also determined was that, oh, hey, adenine and thymine, they seem to go together. That's why they're present in the same amount. Guanine and cytosine, they seem to go together. That's why they're present in the same amount. They're actually paired with each other. And not only are they paired with each other, what that kind of shows is that there is a relationship between the number of purine bases and the number of pyrimidine bases inside of the genetic code. They're equal. They have to. So the pairings that we come up with, A and T, G and C. Again, that's just for DNA. For RNA, same idea, only we're throwing in that uracil, that U, instead of the T. But how does that work? Why is it that way? Well, this is where some more advanced technology was able to help us a little bit. Because what we can see is that the structural similarities between some of the molecules and each other actually allowed for these pairings to occur. So why does adenine and thymine work together? Well, because if you look at their structures, the structure of that purine in adenine lines up pretty well with this pyrimidine in thymine, to the point where I can get these nitrogen and oxygen, nitrogen and nitrogen groups close enough together that they can actually hydrogen bond with each other. Just as a recall, Remember, hydrogen bonding can occur anytime we have polar bonding with oxygen, nitrogen, and hydrogen. So here I have an NH bond getting close to an oxygen. That's a perfect opportunity for hydrogen bonding. Here I have an NH bond on the thymine. It can get close enough to the adenene's nitrogen in that purine group to hydrogen bond with it as well. Adenine and thymine make two hydrogen bonds with each other. And if I look at guanine and cytosine, I see the same thing, only a little bit more. Because now I can see there are 3 interactions possible between the guanine and the cytosine. I can get 3 hydrogen bonds to exist between those 2 pairs. Well, a question you might have for yourself is, well, okay, why? I can see the hydrogen bonding here. Why can't I just take this cytosine, which is also a pyrimidine, and put it here next to the adenine and make hydrogen bonds that way? The problem there is that the way that the cytosine is structured, with that 3rd nitrogen group up here instead of this methyl group, it's clunky. It doesn't fit quite as well together. It's a little bit more boxy. And without that 3rd hydrogen bond there to sustain it, there isn't enough interaction between them to actually keep it together for more than maybe just a couple of seconds or less. So there is a specificity here that is related to these structures. And the fact that the guanine is capable of making 3 hydrogen bonds and the cytosine is also capable of making 3 hydrogen bonds, that's no accident. That's why they're meant to go together. I can't substitute one out for the other because it's not going to work. I can't take something that's meant for 3 hydrogen bonds and put it with something that's meant for 2 hydrogen bonds. The attractions won't be significant enough to keep them together. So now what happens, what we can start to see is that we've got a double helix going here. We've got 2 separate strands of DNA, but they are oriented opposite of each other. So look closely. This strand that ends on the left is actually the strand that is going upward. So remember, we always go from the 5 side to the 3 side. So that strand is going up and around this way. Whereas the other strand, which again, starting from the 5 side and going down, is going the opposite direction. We have two strands of nucleic acids that are running in opposite directions. And not only are they running in opposite directions, they are still aligned to each other. If I have an A on one strand, I'm going to have a T on the opposite strand. If I have a G on one strand, I'm going to have a C on the opposite strand. So that on its own is how we get the ladder for DNA, where every base pair combination, A with T, G with C, in whatever order they're supposed to be in, basically make rungs of a ladder. Where does the helix come in? Well, just like what we saw with proteins. The backbone, that sugar phosphate backbone, is going to be able to do some attracting to itself and cause those twists to take place. And that twisting is going to create a more compact shape than if we just had it as one long ladder. So the end result is a double helix. And the double helix, again, that's the shape that's coming as a result of the complementary pairs, as well as the interactive forces between the phosphates and each other. So keeping all of this in mind, I've got one strand. What would the other strand look like? So what would match the A, the adenine, on this complementary strand? T. What would match the G? C. And the T? A. And the C? G. And the C? And the A? T. And the A? T. And the T? A. And the C? G. So that would be the complementary strand. And again, we would read that strand backwards, right to left. Parker. No, that works. I mean, I went to Grove City High School, so they just told us that everything was at GC. But that only worked for us. Yeah. So, I mean, unfortunately, I don't have a snappy little way to remember that. Yeah, but you know, Parker, thank you for the apples and trees and cars and garages. I like it. All right, last thing for today. DNA. So every time our cells divide, which happens all of the time, regardless of how old we are, our cells are still in the process of replication and division. When our cells divide, our DNA needs to be replicated. Because the genetic information that is held in our DNA needs to be moved to the new cell. And so what happens is that we'll see that the DNA strand is going to be unzipped, so to speak. Now, how it gets unzipped is through an enzyme known as helicase. What the helicase does is it takes that double helix and it starts to pull the strands apart from each other. Now, it doesn't do it all at once. It kind of moves in stages. So the helicase will start on one spot, open up part of the strand, and then what will happen is that different groups will start to bring in the complementary base pair to help replicate the other strand. So the best way to kind of show that is in a picture rather than trying to describe it in long form. And this is kind of what it looks like. So here we can see that the helicase has already done its job in terms of it has started to separate the strands of DNA from each other. And then what we'll see is that zipped apart DNA strand basically serves as a template for new DNA. Since we already have half of the code, then all we need to do is build on the other half of the code. And so we start at the bottom, it unzips, we start to build the code on each side, the helicase moves up, unzips more, builds more up as it goes, until eventually we've copied the whole strand. Once we've copied the whole strand, we've effectively unzipped the whole thing. We now have 2 copies of the DNA. And that's where we're going to stop. Because to go from here, we're going to start talking about RNA and kind of the processes that RNA goes through to take the genetic code. And what's going to happen in RNA is RNA's job is not replication. Replication is the job of DNA and helicase and a couple of other very specific enzymes and proteins. What RNA's job is RNA is going to take the genetic information from the DNA and use that genetic information to start to build amino acid chains. And so it's going to take more than 5 minutes of explanation to do that. So I'm going to leave that for our next class and just start from there. So this is where we will pick up next Tuesday. Don't forget, Thursday, that's our next class. That's also our 4th exam. If any questions come up between now and Thursday, please feel free to shoot me an email or send me a text message, and I'll try to help you out as best I can. Have a good day. Have fun in lab. I'll see you on Thursday.